Welcome to week 3. This week we are going to go over chapters 8 and 9, so 2 PowerPoints essentially for the week, and so we will just jump right into it. It's all about red cell line. So that is the very first cell line that we are going to learn is going to be the erythrocyte or red cell line production. So you're going to see one of the first chapter PowerPoints here. So we'll start with chapter 8 PowerPoint, and then we'll go into chapter 9. So let me just jump into slideshow format. All right, so I'm going to skip through the objectives. Again, you can certainly read those on your own. So with this chapter focusing solely on red cells, first and foremost is what is the function of a red cell. So red cells, of course, carry oxygen from the lung to the tissues, and then they also get rid of CO2 and bring that back to the lungs, and they help with buffering the pH of the blood. There is different namings of our cells that are involved in making the red cells. So sometimes you're going to see the words erythroblast or normoblast in some of the nomenclature or the names of the red cells, just kind of FYI. Anytime the word blast is put in the name, that is referring to a very young, immature cell. So anytime we see blast, you should think very baby, very young cell, doesn't really function yet. It's just starting to be born and made, kind of, if you think of it that way. So with that being said, last week we learned hematopoiesis, stem cell chart kind of deal. So you saw an image of this in your last week's PowerPoint. So we did learn last week, everything comes from that first pluripotential stem cell at the top. And so what we are focusing on here this week is when you go from that pluripotential stem cell to the myeloid stem cell to our progenitor cell CFU gem, the E standing for erythrocyte, and if you follow that line over, we are focusing solely on this erythrocyte line here. So CFU gem will then turn into BFUE, CFUE turns into CFUE, and then finally we have Pronormoblast. Pronormoblast here is going to be the first morphologically identifiable red cell precursor. What that means is that is the first one that we can see under the microscope and identify. So if you were to get somebody's bone marrow sample, because remember all this is taking place in the bone marrow, if you were to get someone's bone marrow sample, you should be able to identify pronormoblast under the microscope from that. Anything before the pronormoblast, so the CFUE, BFUE, CFU gem, all that, we can't see that with a naked eye, with the microscope even. We would need different markers on the surface of those cells to be able to count and know that they're there. So really the only one that we can first see is that pronormoblast. So again, blast is in the name, meaning it is a very young cell. It's not mature yet. It's going to be lots of steps in between there, and then finally it will get to the mature erythrocytes. It actually will take about 18 to 21 days to make a mature red cell from the BFUE stage. So that whole line there on the left can take around 18 to 21 days to make that cell. So as we're making those cells, we're maturing and dividing. Typically from one pronormoblast, you can get about 8 mature red cells made. So how to identify red cell precursors? So you're not going to only need to be able to look at a red cell, you're also going to be able to identify pronormoblast and anything after that. And there's different ways that we can tell each cell apart from. So things that we're going to look at is the size of the nucleus, the chromatin pattern inside that nucleus, the size of the cytoplasm and the coloring of the cytoplasm, if there's nucleoli present or not. As I stated last week, nucleoli are found in young cells. So we'll see that in the first couple as they are young, and then as they mature, we will not see nucleoli anymore. So with red cells, here's what we tend to see in typically. The size of the cell is going to decrease. Overall, most cells start big and get smaller as they mature. The size of the nucleus also decreases, and then you'll gain more cytoplasm. Your nuclear chromatin will become more condensed, your nucleoli disappear, and then finally you'll have your cytoplasm change colors from that basophilic blue to a gray color to finally the pink-red color that we know makes up our red cells. So this is kind of what we're going to see in the red cell line as it matures. So letter A column is showing you how the cell size and the cytoplasm color changes as it matures. So you can see it starts out big, gets much smaller, and then finally turns into that pink kind of color at the bottom. Letter B is showing you the nucleus, how that decreases in size, and then finally we lose the nucleus because your mature red cells have no nucleus. When you look at the microscope, on your peripheral blood, just like you did last week, you'll know that your red cells have no nucleus. There's just the red color with a nice white central pallor, but there's not a nucleus. So we will eventually ditch the nucleus. In letter C column, it's showing you how that chromatin inside the nucleus starts looser, and it will clump up as it goes and become one big solid mass. And then finally, letter D, putting it all together. So this is kind of a pictorial representation of what our cells are going to look like as we mature from the pronormoblast down to the fully mature red cell. So that's what we are going to learn here is this sequence right here. You need to know this in order. So again, the first cell that we can see under the microscope is going to be the pronormoblast. That will mature into the basophilic normoblast, into the polychromatic normoblast, then the orthochromic normoblast, then the reticulocyte, and then finally we will have our mature erythrocyte, or what you can call a red cell. So this is the magical line, and we're going to need to know how to identify each one of these cells along the way. So this is just a nice little chart that kind of summarizes the features of each. I would know this will help you. So like it shows you the overall size of the cell, the nuclear to cytoplasm ratio. So for example, in the pronormoblast line, that nuclear to cytoplasm ratio is 8:1. What that means is it has a very large nucleus and very little cytoplasm. As that nucleus number goes down to like 4:1, that's saying a lot smaller of a nucleus and more cytoplasm then. It tells you if it has nucleoli, you'll notice that only the pronormoblast and the basophilic normoblast have nucleoli. Nothing else does. So those are kind of some features to help us in identifying these cells. So now we actually need to look at pictures because that's the best way to get to know them. So here is a picture of a pronormoblast. So like is shown on the other side, it's a large cell. It's 12 to 20 micrometers. It has that 8:1 nucleus to cytoplasm ratio. Again, meaning that nucleus takes up almost the entire cell. And if you look, it does. There's only a little bit of cytoplasm that's featured around the edges. It has 1 to 2 nucleoli. Now, in this picture, I can't really see the nucleoli very well, but they are there. If I had a microscope, I could probably focus it up and down. And you'll notice that the cytoplasm is still that very dark blue color in this. So everything's very darkly stained, very large. And you know it's a large cell. Even if you don't have a ruler to measure it, you can tell it's a large cell when you compare it to the things around it. So when you look at the other cells in the picture here, you can see these are the regular red cells. They're a lot smaller than this cell. So you know this is a very large cell. And then, of course, just a little note from last week, we had mentioned that this little clear area next to the nucleus, that is the Golgi apparatus, because remember, it doesn't take up stain very well. All right, so here's another look at the pronormoblast. So just another picture. This time I do see it for sure, 1 nucleoli. My mouse pointer is kind of circling it. I hope you can see my mouse pointer. There, it's on the upper top of the nucleus toward noon. But anyway, you can tell again, if you look at this cell compared to everything else in the picture, way larger than everything else. Very dark blue still. The nucleus takes up almost the entire cell still. So that is a pronormoblast. The next line was a basophilic normoblast. It's only 10 to 15 micrometers, so it's slightly smaller. 6:1 nucleus to cytoplasm ratio, meaning the nucleus got smaller. It's still very dark blue. In fact, it's named a basophilic normoblast for how dark blue it can be. The chromatin inside that nucleus, you're starting to see it's a little chunkier looking. And then sometimes it can have up to one nucleoli. It doesn't always have a nucleoli, but it can. So polychromatic normoblast is next. This is smaller yet, 10 to 12 micrometers. And when you look at this cell compared to the normal red cells, you can definitely see it's starting to get almost to that size. It's getting closer. The other big thing you'll notice is the cytoplasm. It's not that basophilic dark blue color anymore. It's a gray. It changed color. Now it's a lighter color. Also, the chromatin inside the nucleus is very chunky. It's what I call a checkerboard clumping. So you know how on a checkerboard it has black squares and white squares and black, white, white. It's kind of that. It's got dark clump, lighter clump, dark clump, lighter clump. So it's very well characteristic to have a checkerboard clumping in the nucleus of a polychromatic. That's what makes it really identifiable. So again, no nucleoli in this. Orthochromic, by far, out of any of them, should be the easiest. And you'll definitely, if out of all of these, you'll see this one the most in the regular blood. So all of these are happening in the bone marrow, but in certain diseases, some of these spill over and come and you can see them in the blood. This is probably the one that you'll see the most commonly in the blood. So erythrochromic normoblasts, you can see again, it's almost the exact same size as the regular red cells. It's just a little bit larger than them. It's still 8 to 10 micrometers. Very small nucleus, more cytoplasm than nucleus. Again, that cytoplasm is a very light gray, and then the key feature is the nucleus. Do you see how it's one solid mass? The chromatin is completely condensed. It's just one big solid mass of a nucleus. And we call that pycnotic. Pycnotic, pycnotic nucleus, or that dense, dark chromatin nucleus. All right, that will mature into a reticulocyte. As you can tell, orthochromic normoblast was the very last stage to have a nucleus. The next step, no nucleus. So that nucleus tends to sit to one side here as it's ready to expel it, and then as you move into the reticulocyte stage, there is no nucleus. Now, the arrows are pointing to the reticulocytes. You'll notice that these are not quite pink-red yet. They're still that light blue-gray color, and that's because they have some RNA left in them, and that RNA still stains blue. So that remnant RNA is what's responsible for keeping them that little bit of a blue coloring. Again, they're slightly larger than our regular red cells still, so they're still about 8 to 10 micrometers. We do have normal amounts of reticulocytes in our bloodstream at all times. So roughly 1 to 2% of your cells are reticulocytes in your bloodstream. So of course, we're doing a lot of our maturation in our bone marrow, but at some point they will put out the retics, and then as they mature into red cells, they'll already be in our bloodstream. So around 1 to 2% is healthy, normal for all of us to have in our blood. I think your book might say 0.5 to 1.5, but I just say 1 to 2 is easier. And then, of course, our mature red cells. So you should have seen these under your looking at peripheral blood smears last week. They look nice and pink, red. A lot of times they're called salmon pink. They're 8 to 8.5 micrometers is the normal size for them. Again, they should have a nice central pallor that takes up about 1/3 of the red cells. So you'll see that in some of them. Not all of them here show a central pallor clearly, but you definitely see that white area, that central pallor. All right, so that was our red cell line that you have to remember. I do ask that you remember it all the way from the start, from pluripotential stem cells down to erythrocyte. Now, the pictures that you need to know were just pronormoblast to erythrocyte. And then you'll notice here there's 2 other naming systems, because in healthcare, we love to triple name everything. I mean, why would we just keep one name? Why not double, triple name it? It's so frustrating. I get it. So unfortunately, you got to know all naming systems. I hate to say that, but your boards could really throw out any name at you, and I wouldn't want you to get the question wrong just because of a stupid name change. So most people, well, at least most people in my world, use the naming system that we just went through, the one that's on the left here, pronormoblast, basophilic normoblast, polychromatic, and so on. That's the one that most people tend to use. Now, if you go to the column all the way to the right, the erythroblastic naming system, it's the same thing. They just replaced the normal with erythro. So instead of a pronormoblast, it's a pro-erythroblast. Basophilic normoblast, it's a basophilic erythroblast. That makes it simple. So those 2 are manageable. It's the darn middle one that's like the weirdest thing. It's so goofy. And it's kind of an older school naming system. I don't hear it used very much. It's kind of more older school text, but maybe it could pop up here and there. So I would just want you to be familiar with it. Make sure you can use these interchangeably. So if we were to say rubroblast, you know that's a pronormoblast. If we were to say prorubrocyte, that's a basophilic normoblast. Then rubrocyte, metarubricite, and then finally the last 2 stay the exact same names. So do memorize the naming systems. Unfortunately, it's just a memorization thing. All right, so a little bit on what we call erythrokinetics, and I mean little. We're just going to define a couple terms that are in this area. So if you ever hear the term erythron, this is just referring to all stages of red cells throughout the body, whereas a red cell mass is only referring to those red cells that are currently in the blood circulation. So that's just the difference. I don't really test on that. It's just FYI. All right, so now in order for red cells to function and carry out oxygen to our tissues and bring back CO2, they must have a whole system in place to know when to carry that oxygen. So there is what we call oxygen sensing cells in the kidneys. They're actually the peritubular interstitial cells in the kidney, whatever. It's just kidney cells. So those kidney cells can sense when your tissues have hypoxia, meaning very little oxygen left in them. When that happens, the kidney cells will produce erythropoietin, EPO, and that will go to the bone marrow and help stimulate and speed up kind of the process of getting more red cells out there. We want to get more red cells out there to carry more oxygen to those tissues. So you need to remember that erythropoietin, we mentioned it last week, that's a hormone, that's a growth factor for the red cells. You need EPO, you need erythropoietin hormone in order to make your red cells. If you don't have it, you'll never make a red cell. So this whole oxygen sensing system is in place in the kidneys to help produce that hormone so that it can go to the bone marrow and get more out there if we need it to. All right, so this slide kind of just defines that. Erythropoietin hormone produced by kidney acts as a growth factor. So here's kind of what some of the things that it can help do. It can help release reticulocytes early from the bone marrow so they get in the blood quick, they can mature into red cells. It allows more cells to mature into red cells than before. It reduces the time needed for them to mature, so it kind of speeds it up. So all of these are things that it can do if we're really getting short on oxygen. Overall, your red cells live 120 days. That is the magic number to remember for red blood cell life. There are two different forms of basically dying of red cells, if you will, the hemolysis. So hemolysis is a term that means breakdown of red blood cells. So there's what we call the macrophage-mediated hemolysis. So anytime your blood is filtering through your spleen, and there are macrophages in your spleen that are kind of scanning it. They're trying to get rid of anything that's foreign, anything that doesn't belong. They're looking for inclusions, things like that. They're also scanning for old, dying red cells. When they see them, they eat them up and take them out of circulation. So that's probably the more common way that our cells die off. So as they reach that 120-day lifespan, typically it's those macrophages that help take care of them and get them out of the circulation then. The other means to which cells may die is mechanical hemolysis. So this could be in an infection, a drug that you took, something that you put in your system that's all of a sudden causing your cells to lyse when they shouldn't be lysing. But that does happen. All right, so that was chapter 8, a lot of information. You have to know those pictures and the characteristics of each of those cells in the red cell production line. That will be really important. They'll be on an exam coming up soon. All right, now let's go over to chapter 9, which is metabolism of the erythrocyte. We're gonna just skip through the objectives. So again, mature red cells do not have a nucleus. They do not have organelles. They don't have the normal stuff that younger cells do. So as a result of missing the nucleus and organelles, they don't make anything. Mature red cells can't make proteins, can't make lipids. They have to get their energy somewhere. So in order for the red cell to get an energy so that they can perform their function, deliver oxygen, they need to go through kind of what we call a glycolytic pathway. So they also need to have an intact red cell membrane, and I'll mention that there. But basically, anytime we think of energy, you should be thinking of ATP. ATP just equals energy units. You can also know it as ADP or AMP. But essentially, by going through this glycolytic pathway or glycolysis, they will convert glucose, remember glucose is energy, into pyruvate with the production of ATP. So what that means is this will equal getting them energy by converting glucose into energy. And you can do this either aerobically or anaerobically. So anaerobic is the main source of energy for our red cells. It is responsible for 90 to 95% of glucose consumption, and it's taking place in what we call the Embden-Meyerhof pathway. Now, I don't go a lot into depth on this pathway. If you look in your chapter, it's kind of overwhelming. To me, that would be more like if you got into a bachelor's degree level, they go more into this. For our purposes, we're going to keep this really light, and what you should know is that Embden-Meyerhof, which is a form of anaerobic glycolysis, is responsible for the majority of energy, and what it gives us at the very end of that pathway is 2 molecules of ATP. So it's going to give us 2 ATP units of energy at the end of that anaerobic glycolysis pathway. There is another pathway that's called the methemoglobin reductase pathway. Methemoglobin pathway is all about keeping our iron maintained in the state of 2+, in that reduced 2+ state. So I know that we haven't learned our hemoglobin molecule information yet. We're going to learn it next week. But for those that have forgotten, maybe you didn't get this in A&P as much, inside every red cell is hemoglobin. Inside the hemoglobin is iron. You have to have iron there to help bind oxygen to deliver it. Iron has to be in a 2+ state in order to bind and deliver oxygen. You need it in 2+ state. This whole pathway will make sure that it stays in that 2+ state and doesn't change so that you can continually bind and transport oxygen. That's it. That's all you need to know on that for now. All right, the other form of glycolysis was called aerobic glycolysis. There is a pathway under this called the hexose monophosphate pathway, and it's bolded because I find it important. The whole purpose of this pathway is to convert the glucose into what we call eventually glutathione. So glutathione is a protector. It's a bouncer for your cells. It's going to protect your cells from oxidative injury. In fact, you can see this as part of some supplements. If you ever take supplements or know somebody who's into health and fitness, they might talk about glutathione a little bit. It's something that has become a little bit more popular. But we naturally have this in us anytime that we go through this hexose monophosphate pathway. It's to get us that glutathione, and that glutathione is just going to keep your cells safe, protect your cells from oxidative injury. And there are different drugs, different things that you could be exposed to that would cause oxidative injury to your cells. So that's the whole purpose of hexose monophosphate pathway, just to get us glutathione to protect our cells. All right, so overall, the red cell membrane serves itself a huge purpose in allowing the cell shape to be maintained, to protect the cell, keep it whole, fit it through areas. So your red cell overall is called a biconcave shape. That biconcave shape just gives flexibility, helps change substances from the outside to the inside, that kind of deal. So it will also help with having a good skeletal system to make sure the surface antigens and receptors are there. So there's different pathways, transportation pathways like passive and active to get stuff in cells. I am not going to ask you questions on that. That right now we don't need. That probably was more basic anatomy and physiology kind of stuff. All right, but overall, your red cell membrane is what we call a lipid bilayer. It's made up of phospholipids, 40%, and 52% protein. So it's kind of that double lipid bilayer, and then proteins are embedded into that, and then there's a little bit of carbohydrates mixed in. That is it for metabolism of the erythrocyte. That is a really complicated chapter, and we have just basically done is whittled it down to the essentials, the things that I really need you to know on. And those are just the pieces that allow these cells to function and keep going. So if you have questions, as always, please feel free to reach out. Otherwise, I hope everybody has a fabulous week. Thanks.